Okay, we're going to get started. Thanks everybody for coming. So this is part 2 of the deposition of mercury and effects of mercury in toxics. And I'm Colin Eaglesmith with USGS.
I'm coughing. David Schmeltz with EPA.
We're going to be the session chairs for you today. So our first speaker is Ryan Lepak with US EPA, and he's going to be talking about understanding spatial atmospheric mercury deposition to aquatic ecosystems and the landscape surrounding using National Lakes Assessment. So take it away, Ryan.
Great. So this is a project that's quite long in the tooth, and if I'm being quite frank, was kind of born out of the ridiculous aspirations of an overzealous graduate student. But since then, that graduate student's gone to the EPA and has made earnest efforts to try to rectify this problem. So I'm Ryan. I'm working with a team of people at EPA, including Kelsey Vitens, folks at Harvard, and then of course the analyses were all performed at the Mercury lab here in Madison, the USGS lab. Tremendous amount of appreciation for everyone involved because this is one of those community efforts. I am not an expert in almost any of the things I'm talking about today. So the backdrop of this in the perspective of ORD is we're kind of obligated to do some sort of monitoring or evaluation of the Minamata Convention on Mercury. And there's been, kind of published successes that we've already heard about, including massive reductions in mercury load or emissions, largely tied to power generation. And we're seeing that in the air. We're seeing concentrations in the atmosphere just declining kind of at a steady pace. And the responsiveness to those declines is kind of variable, and it really depends a lot on the landscape. It's super complex, and so we thought at the time, let's measure mercury isotopes everywhere in the air, and we'll be able to kind of seek out these sources very easily and understand, the city of New York and its influence, et cetera. And what we learned from that exercise is it's very good at finding massive mercury sources, but when we try to look at the regional level, we can only do just that, very regional-based trends. And so it kind of led us to kind of think, well, what other spatial indicators might we learn to better evaluate the mercury system as it's depositing to the landscape, as it deposits to the lakes? What are the things that are mattering, and how well can we use things like mercury stable isotopes to identify sources? So a little backdrop for those who don't know, it's very brief. The National Lake Assessment is what you might guess coming from EPA. It's a statistical survey of the condition of our lakes. This is not intended to be a mercury-based project. It's kind of this whole atmosphere of information we're trying to glean from that. Things like biological condition, recreational status, major stressors to those lakes. Here we're using 2012 lakes, again, long in the tooth, so I apologize. It's a randomized design, and they've been doing this kind of semi-frequently since 2007, as recent as 2022. And a lot of this is, the impetus to do this has ties to the Clean Water Act, some of our EPA objectives, including those at ORD I just mentioned, and then just tying to the NARS program as a whole. And this is old, and I'm only going to present the 0 to 2 centimeter data. It began when we were still in Middleton, and that move happened in 2019. Now we're just right down at the Forest Products Lab. We're talking about a lot of samples here. Just for concentrations, you can kind of see some of the data. We got over 1,000 lakes for concentrations we've measured. And a subset of those, a statistically comparable subset of those, we measured for mercury isotopes, completely done gratis. This is an exploration for our science, for us to learn better about what we can do with this tool and to ground truth the things we can and cannot say using mercury stable isotopes. So traditionally, how I'd present this data, if it was just me and I didn't have this wonderful team behind me helping me do things, is I'd say, we've measured concentrations across the US. We see that there's this huge range, and a lot of the range in the concentration is really just tied to the amount of carbon in the sediment. And most of you didn't learn anything. And I can do the same thing with methylmercury. If you have more total mercury supply, you probably have more methylmercury created, and again, no one's learning too much. And the percent of that methylmercury sometimes is tied to how much organic carbon is in the sediment. That all makes sense to you guys. And if we measured isotopes, I'd start to tell you, we kind of measure this continuum of values across the landscape. We've got some that are very low and some that are very high. And if we try to linearize it to talk about processes that mercury itself is undergoing, we could certainly do that and talk about the importance of photochemistry, the importance of tropopausal inputs of mercury, et cetera. We can validate how good we did. We do just fine measuring these things. And we can take these associations, try to learn something just simply about the mercury cycle explicitly, but we're gonna try not to do that today. Those things do work. I could go and create a mixing model and say, okay, some proportion of these lakes are more like a watershed. We use these end members. Some are more like precipitation, some like industry. And if we wanna get really fancy, we could put out some sort of mixing plots for you guys. So something you might have seen before, if not, you'll see in the future. Just kind of what are the kind of pathways of deposition to any given ecosystem. That's how we've done mercury science in the past. We're going to try something different. So to do this, we built this team. First up for this team is Benjamin Gaiman at Harvard, and he's our modeling expert as it pertains to the geochem model. So he was able to build us specific models for the time zone of interest, so let's say 5 years preceding 2012, and then he built very small grid cells so we get a little bit more precise data. He did this across the landscape and also vertically for us. And the things that we got out of that model for every site were large-scale wet depth, convective wet depth, dry depth, surface concentrations, et cetera. So we take that information, we raster it against the sites we have. And then we got this team of GIS specialists, some at EPA, like Tom, the top left, Brandon Crumwade in the center, and then Mike here at the bottom right. And they started kind of diving into some of the eco-region level information and trying to pull out some of the stuff that exists in random data sets across the US, including NADP, looking at concentrations, depositions there, and lake cat data sets, et cetera. We want to look at nitrogen deposition, land use characteristics, catchment characteristics. We're trying to pack everything into this. And then comes the real special sauce to this for us is the person able to do a lot of this random forest modeling. This is Kelsey, she's at the EPA. And I'm not going to belabor the topic of random forest because frankly I don't understand it all that well. What I do know is that it's this iterative process and the way she's designing it has a lot of bootstrapping in it. So we're talking about analyses upon analyses and redundance over and over and over, rejecting things that don't seem to predict error. And that's kind of how this process goes. So you think of something like carbon content or LOI in sediment. You can say, okay, well, what drives the carbon content across the landscape? Well, the first thing is actually temperature. Now that could be confounded with other things like latitude. And then there's precipitation, and then the catchment characteristics, the nutrient deposition, the catchment composition, hydrologic regimes. I think you guys all expected these things, but this is kind of just a proof of concept for you. Spit it out of those models, we can get a lot of things that are kind of qualitative in nature. We can look at biplots that are partial dependent. So you could say, well, sure, as methylmercury concentrations go up, LOI percents are going up. Or we can do these in a three-dimensional context. As LOI is going up and supply of mercury is going up, you see in the top right a little more methylmercury formation. These things make sense, but these are the qualitative things we can pull out of this type of technique. So we're just jumping into results. I'm not going to tell you how mercury isotopes work. I'm not going to tell you how the model works. We're just going to jump into the results because it's more fun, and we can have questions afterwards if you're interested in those things. Starting with total concentration across the landscape, a couple of things. So she ran all these models in here. She found out what are the major predictors for concentration. Again, it's what you expect. It's the carbon, it's the pH, it's the rainfall, it's the runoff. The geochem model is very important to understand these concentrations. That did surprise me. And then just the wet deposition, origin, and the catchment characteristics once again. Spatially, if you look across the US, if you see more reds or more blues in a consistent region, you would start to see that there's some sort of spatial autocorrelation. We're not seeing much of that, except maybe for California. And I think some of you might know why that is if you know anything about mercury. A lot of gold mining there, where they use mercury. Looking at methylmercury, the same thing. There's no real spatial autocorrelation, so that's good. That means we're doing an okay job spatially of predicting these things. And then when we try to think about what the errors are of our predictions, they're on the log sense, so they're fold predictions. So for total mercury, for instance, if it's a 1.45 log error, that means if you have a 100 ppb sediment, your prediction error is about 50 ppb on top of that, but if you have 1,000, it's about 500. So we're kind of saying it's more of a yes or no science. Is there a lot of mercury, or is it pretty low? And that's what we're kind of doing with that. Same with the methyl. The error is a little bit higher, as you might expect. On these maps, when you do look at these maps, if they're red, like in California, that means it overpredicts. That means the concentrations in the real sense are higher than what we would have predicted. And if they're blue, that means that we overpredicted, and so the real concentration was lower. And that's going to be consistent for all the rest of these slides. But really, we're going to talk about this in the context of isotopes. So here, for the sake of brevity, the listed predictors are for all three isotopes in concert together. But then I've also highlighted in green, orange, and blue the very major predictors for every given single isotope system. So it's a lot to take in, but the take home is pretty much the same. Precipitation is really important to some of these values. Runoff is important to 2 of these values. The geochem is important to 2 of these values. LOI becomes really important to this tropopausal thing. That could be an autocorrelation, we're not quite sure yet. And again, the other surprising one was maybe the degree of deforestation is kind of becoming prominent for 200. So these are things, I didn't give you the background or the context of what these isotope values mean, but just know that 200 is something coming from the tropopause, and we're not quite sure how to use it right now. And so we're trying to build out kind of weight of evidence inferences to understand those things across the landscape. And so it's kind of exciting for the Mercury community. The prediction errors for the isotope values, because we have to get there to actually use these models for anything useful. To read these things, you see on the y-axis, you got the predicted values that we would have done across the landscape for the ones we measured. And then in the bottom, you'll see the measured ones. And the output of that, what that means is the prediction error for any given lake is something like 0.14. And maybe that doesn't mean a lot, but let me tell you this. When I measure this same isotope, our analytical uncertainty is 0.06. So we're just about double the analytical uncertainty for some of these isotope values. And then if we bring up the other ones, they're also comparable. They're pretty low abilities to predict these things, or very high certainties rather. And we also held out 34 lakes just to run those separately, completely independent of the model, ran those, and the results were actually the same. That's where the RMSE came from. The MAE was innate to the model itself. And again, we got a little bit of spatial autocorrelation, not too much, and that may mean that something could be improved, or that could be a factor related to just there's areas where there's sources. I point you again to California and look at the 202. So that's right here on your left. You see how there's a bunch of red clusters. That could just simply be forcing from the gold mining. And you see a couple blues here and there and reds, et cetera. But it's still minor to moderate spatial autocorrelation, meaning we could maybe improve something, or there's just something unique to that area that isn't being captured with our modeling technique. So what's the purpose of doing all this? Well, we want to take that information that from the 400 we measured and extend it to the 1,000 we have, or any other lake that we have those characteristics for, so that we can kind of create the US's first isoscape. Now, when I first set this up, I was thinking, well, we might have the ability to make something very coherent where we can very easily say, okay, this region's always this region's always that. And sometimes that's true. Like, for instance, the Northeast is consistently a lower value. But in some areas, it's a little more variable across, especially the central US. So this is 202. This is 199. And this is 200. So in the Mercury world, we would think about the 202 as being sources. If I saw big plumes one way or the other across the landscape, I would say, well, there might be something source-related going on there, or something sorption-based related going on there. That was the kind of the old hat thought process on how we do this. But you saw what the predictors were. The predictors were actually much more climatic in scale, land use in scale, rain events, et cetera. We're not so much looking for, or we're not showing precise source deposit areas. It's more something mechanistic related to the ecosystem characteristics. That holds true for 199 and 200. Now we'll kind of tip our hand what some of these things mean. 199, as that value goes up, the prominence of photochemistry on the Mercury prior to deposition, or while in the lake prior to sedimentation, has gone up. Now, we all know Duluth isn't getting more light than down here, so that's not something to do with more light shining. There must be something either geochemical or some other unique predisposition. Likewise with the 200, higher values mean that the tropopause-related rain mercury is of a greater proportion. And so for some reason, that value is getting more prominent in the Minnesota area, the Wisconsin area. We haven't unpacked why. I've seen some of the models earlier today in the earlier session. I saw a couple spots where ammonia was a little hotter there too. Interesting to see if we could kind of take some of these models and raster them against each other another day. The end of the day, we're trying to create, like I said, these isoscapes so that we could really more concretely pin something to a geolocation, right? So for instance, they use this a lot in migrational patterns, right? If a bird has a delta value in hydrogen that's very high, it maybe came from the south and it's migrating north. We were hoping to do something along those lines. Instead, what we learned is its ability to take any given ecosystem and see how it either agrees with or doesn't agree with the norm. And if it doesn't agree with what we predict, maybe there's something unique going on in that ecosystem. So some of you may have been outside, and there's two lakes right around here. We can kind of play this game if we want to. And we look at present-day Mendota, which is to the north, and present-day Monona, which is to the south. And you see the values are about negative 1.06, negative 0.45, and those two fall in very differing areas of my little mixing plot that I have on the right. This is an example. We predicted Mendota very well. We predicted Monona very poorly. Well, Monona happens to have in its watershed a major mercury source. Let's step back in time, and that's what the red triangles are, when the mercury source is actually prevalent in both lakes, and now you see those values fall right back to the industry for both lakes. So again, in the past, this model would have predicted Mendota poorly. So what are the kind of qualitative things that this room might get excited about? We think about it in the context of the context of climate change and the precipitation runoff that might follow. Think about it in terms of inputs. You get more precipitation, you get more runoff, you're going to get more total mercury loading. That makes sense. I shouldn't have to convince anybody of that. But if you have more runoff and you have more precipitation, you don't necessarily have more methylmercury production. That's what the red is here, methylmercury as a ratio of total. So it's not that you're just uniformly creating more methylmercury in the ecosystem because you added more. And even if you make it more cloudy, if you decrease the water clarity, it doesn't necessarily mean that you're creating more methylmercury. Instead, it's the important part of this is what those particles are that creates that lessening of Secchi. And as productivity starts to jazz, all of a sudden methylmercury production starts to go up. And so that's very important to know that as we kind of become more increasingly eutrophic, there may be instances where more methylmercury is being produced. So that's kind of like a big picture climate change thing that just fell out of this data set across the US. Spatial drivers. So I pointed very diligently to the Midwest area as kind of this unique thing happening, especially kind of North Dakota, Minnesota, Wisconsin. If it's not from the atmosphere, could it be something from the glacial lines? There's some evidence because a lot of these values in that area, when I just take that area and I analyze only that one, there's a lot of linearization going on with how much iron's in the catchment. So that's really interesting. Could be somehow related to that. Also, one of the major US sources persists in Montana still today. So it could be downstream. I don't tend to think it is, but it could be a downstream effect as well. And then you look at the coast. You may have caught that kind of by Colin's hometown. And you're going to know this way better than I am. But there's these isotope variances in that one little sliver along the coast. And I surmise, and I could be wrong, you can correct me, that this could be halide scavenging of the coastal waters that are depositing mercury in a different way than the rest of the US. And that's why you see different isotope values. And that's also why you probably see these big red lines right down the coast. So now we've just added a more concrete mechanism on the national scale to that little zone of the US. Now you guys knew this, but you've never seen it in an isotope way. So where can we go with this? Beyond mercury supply, it seems like the ecosystem-related factors are largely regulating across the national landscape the isotope values we measure. And so one thing we could do is, Minimont is trying to do this monitoring, is revisit this in 2012, right? We did this 10 years ago already. These sediments are 10 years old. What's it look like in 2022 or 5 years from now when they do the next one? Office of Water is very interested in understanding biological responsiveness across the landscape. Of course, we can't do that right now because we're devoid of the data, but could we use this similar kind of learned approach to measure a series of BAFs across the landscape, predict what drives those BAFs, and then set mercury baselines for bioaccumulation across the land? That would help Office of Water understand baseline mercury sensitivity to any given mercury. Temporally, ecosystem, ecological, climate factors influence the concentrations we measure in these fish, and often they're highly variable. And a lot of that variation can be constrained by things like ecological factors. But a lot of times, our ability to measure things are dependent on what was collected in the past. So can we hindcast things using this kind of stuff to, like, for instance, hindcast age on fishes we collected in 1978, because the EPA has been collecting fish since 1978. And if we can we create a mercury model that includes age and all these other cofactors to understand where mercury concentrations are going and then project them under different climate scenarios? That's something you can do with this kind of technology. The other avenues, this extends well beyond mercury. People have published this in nutrient worlds, volatile organic contaminants, and you guys saw the talk with Ross, black carbon. Some of those things could be part of this as well. And I'm personally motivated to try doing this using some carbon nitrogen stabilized tips because I think there's an ability, not unlike the oceans, to create a kind of independent baseline assessment of lakes across the US just using the catchment characteristics and say, okay, we expect baseline carbon, baseline nitrogen, isotopes look like this, and nutrients look like that. That's where we want to go with this. That's not where we are. This is the beginning. And I thank you and take questions.
Thanks, Ryan. We've got time for one or two questions.
Now I'm in trouble.
So Ryan, how do you think this will work for streams and river systems if you ever expand into those?
Yes. No, I think that's, yeah, I knew it. So it depends on where you want to go. I think streams and rivers will be a lot harder, personally. If you focus on headwater, it'd be easier, but there's so many things. There's so many points of inputs that I think it would likely fail unless you start to account for those things. And so you maybe would just have to take it a little slower than what we did. We kind of, even though this is 10 years old, we've done most of this work in the last 3 months. So, yep.
Any other questions?
not so much a question, but just a heads up. On the 2022 NLA, I was part of a group that did 250 lakes across the northern lakes and forests. And so, covering that area where you have that kind of anomaly. And it's being run for isotopes, not mercury, but so your ID you just mentioned may have some applicability, so.
Yeah, that's great, especially if we can channel in there and find instances where there's yeses and nos, because right now we mostly are only found yeses of high 200s, but if we can find nos, we've got something to run against to start to really validate these and remove the spatial autocorrelation issue.
Okay, we're out of time.
Thank you for your time, guys.
Thank you, Ryan. And next up, we have Vivian Taylor with Dartmouth College, and she's going to be talking about some of her work in Lake Champlain.
And I might need help setting this up.
I'm just going to screw this up, but I'll try.
All right, great. Oh, I did it. And then this.
You're not up here, though.
What do I do? I don't know.
Oh, there we go. We're good.
Oh, all right, cool. Thank you. All right. Oh, now what do I do? Display settings. Any guesses? Second one. All right, cool, thanks. Oh, all right, we're good. Thank you. All right, so it's nice to be here, and it's really great to give this talk right after Sarah, because I'm going to be touching on some of the great things, the same things. So we are in a good place. All right, so I'm going to be talking about the Lake Champlain region. So Lake Champlain is a large lake, not a great lake, 120 miles long and 400 feet deep, and it's actually in the same St. Lawrence ecoregion as the Laurentian Great Lakes, so a good paired study. Unlike the Laurentian Great Lakes, though, Lake Champlain has a much larger watershed to lake ratio of about 18 or 19 to 1, whereas the Great Lakes are sort of more like 2 to 1 or 3 to 1, and so it has a much bigger watershed influence. It also supports a very large recreational fishery. There are bass derbies almost every weekend on the lake. And mercury, unfortunately, in those game fish often exceed EPA criterions calculated for human health. Lake Champlain is also the location of one of the very first Mercury Deposition Network sites, the site in Underhill, Vermont, and that site has shown an almost significant declining trend in mercury flux and precipitation over the last couple of decades. And that's important to this study because in the last few years, we have not really seen any big changes in mercury deposition. And so again, what's happening in the biota has nothing to do with what's happening in deposition. But there are, of course, other changes, environmental changes, which can have a big influence on mercury bioaccumulation. And so Lake Champlain, like much of the Northeast, is under intense climate pressure. The average August surface water temperatures in the lake have increased about 7 degrees since 1964. We're seeing increases in nutrient loading, which are driving massive cyanobacteria blooms, particularly in the shallow basins of the lake. And then we're seeing this increased storm intensity, which has caused major flooding events, particularly in 2011 and 2023. And so you can see from the flood in 2023 that those events release a lot of sediment along with nutrients and wastewater that's being rinsed down from the tributaries into the lake. And that can have that can cause an ecosystem shift. So Lake Champlain is also a really interesting lake to study for mercury. Like the Great Lakes, it has a high pH and low DOC and these very low dissolved methylmercury concentrations in the water column. And we also see this very efficient bioaccumulation where the bioaccumulation factors between the dissolved and the water column into the phytoplankton are around 10 to the 6, so very efficient bioaccumulation, and then trophic transfer into the zooplankton is also quite efficient. And what that means is that very small changes in the water column can have a big effect higher in fish high up in the high trophic level fish. It's also interesting because the lake consists of 7 adjoining basins, and those basins have slightly different trophic statuses. So while they're connected, they're sort of cut off from each other hydrologically. So the main lake basins are the deep basins, the North Main, and South Main, which run north to south. And then over on the east, we have Missisquoi up in the northeast corner. That's a very shallow basin. It's about 5 meters deep, mostly river influenced, and that one's really susceptible to eutrophication, as is St. Albans Islands, which is in the northeast arm. And then Mallett's Bay is a bit of a different story. It's also anthropogenically influenced because they put there's a railway bridge that goes between Mallett's Bay and the main lake, and then another highway that goes from the mainland over to the Lake Champlain Islands, and that sort of cut off Mallett's Bay hydrologically from the rest of the lake. It's a really deep basin, and it's started to stratify, so that basin's actually become oligotrophic. And so that gives us this pattern of different trophic status by basin, which allows us to look at bioaccumulation between basins in the lake and how those basins respond to climate change. So over the last couple of years, we've conducted a spatial and temporal study of mercury bioaccumulation. The Lake Champlain Basin Program, which is our funding agency, funds a game fish mercury survey every 5 years since about the 1980s, and this has become a really good record of mercury in fish. And so we conducted the study in 2022, and it's quite a big study. We were asked to collect 10 individuals of each species per basin in each of the 7 basins of the lake. Those included white perch, yellow perch, smallmouth bass, walleye, and lake trout. Lake trout are a cold, deep water species, and so we're only finding those in the main lake. And then in 2023, we did a study of the water, the cestin, and the zooplankton across those different basins of the lake. And so we were looking spatially, both horizontally across the different basins of the lake, and also at differences between the hypolimnion and the epilimnion in the main lake basins. So when we're looking at fish data, this is all mercury data in yellow perch from the last, from all of the surveys, broken up by year. In the earlier years, there was a fairly scant amount of data, so we kind of grouped some of those together. And we always have this covariance with fish size, and so we have to do a covariant analysis to take out the effects of size on fish mercury levels. But what we see on the left are the least squared means of mercury over time, over these different surveys. And we see quite a significant decrease in mercury concentrations in fish from 1988 down to 2011. And then we see a striking increase in 2017, where concentrations this is all yellow perch from all basins of the lake, increased in 2017. But then in our survey in 2022, concentrations came back down. There's always one species that doesn't act like all the other species, and so I just had to throw that in, which is the white perch. Those are more benthic feeders, and they do not have the same temporal trend as the other fish. We haven't analyzed our smallmouth bass yet, but the smallmouth bass, up until 2017, the trend looks very similar to the yellow perch. So it does seem to be an ecosystem-wide, or that we're seeing the same trend over a number of species. And so lake trout, which are much longer-lived cold water fish, also seem to show this trend where there was a big decrease down until 2011 and then a really large increase in mean concentration in 2017. And it seems to be coming back down in 2022. I will say that we're catching most of our big fish from fishing derbies, and so we have like an unfortunately large size distribution, which makes the statistics a little bit difficult, but I've broken this up a number of ways, and I do think the trend holds. So when we break down this temporal trend by basin, it actually turns out that most of that trend of increase from 2011 to 2017 and then back down in 2022 is occurring only in the deep main lake basins. We're not really seeing much of a temporal trend in the other basins. And then I'll just mention that spatially, so within 1 year and across basins, we often see that Mallett's Bay, which is the oligotrophic basin, has the highest mercury fish concentrations, whereas the more eutrophic basins, the Missisquoi doesn't really show up here, but the northeast arm generally have a little bit lower mercury concentrations, as we would expect from the quenching effect of these phytoplankton blooms on mercury bioaccumulation. White perch, again, absolutely no trend. So what causes that big peak in 2017 in fish mercury concentrations? The lake does support a long-term monitoring program that's quite expansive. They do water quality monitoring every 2 weeks during the summer, as well as plankton and density and taxa monitoring. And in looking at it, there's not really a smoking gun. So it's difficult to, this fish data, unlike what Sarah showed, we're only seeing fish mercury concentrations every 5 years. So we have, 2011 and then 2017 and then 2022. And so it's not really clear what happened in those in-between years, like did mercury go up a lot in 2012 and then start to come back down, which makes it sort of hard to know what to look for. One thing that did happen was in 2011, there was the first major storm was Tropical Storm Irene, which delivered massive amounts of sediment along with nutrients and organic matter to the lake. And this is one of the first times that we've ever seen turbidity reach the deep basins of the lake. And so the mechanism by which that would then cause higher methylmercury bioaccumulation is a little unclear, but there is this sudden delivery of organic matter and sediment in the deep basins, which may have caused some sort of an equilibrium shift. So in 2023, we did our water column and zooplankton sampling and analyses. And so this was in, we looked at Mallett's Bay, the 2 shallow basins, and then the 3 main lake basins in both the surface and the deep layers of the lake, and at 3 time points. And so when you do a study like this and we're trying to understand a process, you sort of hope for a representative sampling year, and that's really not what we got. We had this massive storm in the middle of July where 7 inches of rain hit the lake and caused major stage flooding in all of the tributaries. And then the lake levels, which is on the top here, remained quite high throughout the rest of the summer and different from previous years. But what we found was that so this is looking at epilimnion dissolved in cestin methylmercury across sites. In the dissolved phase, not any big differences across sites in methylmercury concentration. In July, when the big storm happened, we were sampling a couple weeks later when there was a significant phytoplankton bloom. TSS here is probably a good proxy for chlorophyll. And so in July, there was a big chlorophyll bloom, and then it sort of came back down in most of the lake basins in August, with the exception of the very shallow basins, which really turned into a big cyanobacteria bloom. But concentrations in the dissolved didn't really change in the cestin. Methylmercury concentrations did decline from June until July, and then they stayed sort of low. And that's just a picture of the one of the cyano blooms in, I think that was St. Albans Bay. And so there we have this big increase in chlorophyll, or TSS, and a decrease in methylmercury concentration in the cestin. And so how that plays out in zooplankton, in pale blue we have the deep basins, and like the cestin, we see a decrease in methylmercury concentration from June until July, and then concentrations start to increase in August. The trend in the shallow basins and in mallets is a little less obvious, and partly our bacteria, and so our samples are, I think that really low value for Missisco is probably because it wasn't really zooplankton. So looking at the main lake basins, and if there are any trends in depth, the main lake stratifies thermally over the course of the summer, but it usually doesn't, there's usually no oxygen depletion. So we're not seeing hypoxia in any of the main lake basins. It's pretty well mixed, and that's usually the case. We do see then the chlorophyll bloom was pretty low in June and then got quite large in July and came back down in August. So for methylmercury and total mercury profiles in the water column, we were sort of expecting that we might see higher methylmercury in the lower or the bottom waters of the lake, and that was in fact not the case. There was not much of a trend. So looking at that just a different way, this is methylmercury in the epilimnion and hypolimnion. There's not much difference in the water column between those 2 layers. So again, the chlorophyll or the TSS increases in June, but only in the epilimnion. We don't really see the chlorophyll in the bottom waters of the lake. But we don't really see that bloom dilution, or we don't see a big difference in cestin mercury concentrations between the bottom waters and the epilimnion, which was a bit surprising. We were sort of expecting to see higher concentrations in the bottom waters. So the zooplankton, though, show a completely different trend. So for zooplankton, mercury concentrations increased in July, whereas for the Ceston, they had slightly decreased. And so zooplankton concentrations in the hypolimnion seem to increase in July and then continue to increase into August, whereas the epilimnion has a much lower concentrations in both July and August. So it seems like we're seeing a difference in trophic transfer. We're not seeing a bloom dilution. And so the reasons for that are, again, like it seems like the hypolimnion is particularly sensitive to mercury bioaccumulation. It's possible that we have, or it's likely that we have quite different zooplankton taxonomy between those two samples, and that's something that we'll really have to look into. Different sizes and different taxa of zooplankton have very different efficiencies of bioaccumulation. And then we also have some of the mysids, which are larger, more predatory species, which are present in the lower lake of the main lake basins. The trophic transfer may be caused by a lower abundance of food, but a higher food quality in the lower waters. The blooms that we're seeing are cyanobacteria, and those are not a great food source, and so it's possible that we're just seeing more efficient feeding in the bottom waters. And it seems like the hypolimnion is not affected by bloom dynamics. So in summary, biota don't follow abiotic mercury trends. Different regions of the lake have different susceptibility to mercury bioaccumulation responses to climate change pressures. The bottom waters of the deep basins of the lake are a sensitive region of mercury bioaccumulation, and that's particularly important because that's where all of the fishery, that's where all of the fish that people consume are coming from, the lake trout and the white fish. And so it's particularly relevant to what people are eating. And I have like a million people to thank, but everyone knows Celia, and Saul helped me with all the songs, and Ethan helped me learn to catch fish. So that's it. I will take questions.
All right, thank you, Vivian. Great talk. We have some time for questions, if anybody has any. So I have a question about the deep basin lakes. So they stratify.
They stratify thermally.
Thermally, but
Yeah.
not
No
geo.
oxygen depletion.
And are there climate change predictions for how that might change?
Yeah, anoxia is expected to go longer. We're seeing a significant warming trend in the bottom waters as well as the surface waters, and it does seem like the period of anoxia is getting to be, or the period of stratification is longer.
Okay.
I don't know if we're going to start seeing anoxia.
Yeah, okay. And do they, when it turns over, is it just a single mixing event, or is it polymictic?
No, I mean, the shallow basins which do stratify, every time there's a big windstorm, they turn over. So it's not for the whole summer ever.
Okay.
Yeah.
Thanks.
Thank you. I was curious about the stratification in terms of the mercury, and is it well mixed throughout the water column, or are there significant concentration differences?
No, we really don't see we don't see profile or depth profiles for mercury. We don't see any changes with depth.
I have another question. So you talked about zooplankton taxonomy. Do you know, is there a shift over time? So you had that difference in concentration, and part of it is, you talked about taxonomy. Do the populations change seasonally?
That's a really good question. Like, we haven't finished our taxonomy data, but I guess I could know that from past years. I don't think there's a huge shift in species, but I'm not sure.
Okay. Thank you. All right. Well, thank you very much, Vivian. Great talk. All right, we're going to transition from mercury in big lakes to mercury in big mountains. And Hannah Miller with University of Colorado Boulder is going to be talking about mercury in high elevation wetlands.
Okay, hi everyone. It's good to be here. This project's a bit smaller than the projects that have been presented on so far, but I hope you still get some good information out of it. I am a graduate student at CU Boulder, and I'll share today some of the work I've been doing looking at mercury cycling in the Colorado Rocky Mountains. So the overarching question that's been driving my dissertation research is how does mercury cycle in mountain ecosystems, and what are the implications of atmospheric deposition and climate change on those processes? And today I'll be focusing on 3 questions. First, do mountain wetlands act as hotspots for total mercury storage and methyl mercury production? Second, is methyl mercury exported downstream from mountain wetland ecosystems? And third, what is the impact of methyl mercury production on local wildlife? I'm using Niwot Ridge Long-Term Ecological Research Site in Boulder County, Colorado for my field site. And this is an interesting region to be doing this research, as despite decreasing mercury emissions in the US, which has led to generally decreasing deposition, particularly in the eastern US, there are areas in the west that are experiencing insignificant or increasing trends in mercury deposition, likely as the elevated global pool of mercury, and then also some direct transport directly from East Asian industrial practices. And the Rocky Mountains are a region where we are seeing these significant increasing trends over the past several decades, although some of the more recent trends are dropping off. And especially notable is that the higher elevation sites generally have higher rates of deposition, so those are the dark blue circles compared to the lower elevation sites, the light blue triangles. So this suggests that these mountain ecosystems may be sensitive to atmospheric mercury deposition, especially because they receive high rates of atmospheric deposition through rain and snow, higher than the lower elevation regions, and then they also have aquatic regions on the landscape with conditions conducive for microbial conversion of inorganic mercury into methyl mercury. Mountain regions are experiencing disproportionate impacts of global change as well, which potentially could impact methyl mercury production in these areas. Long-term data from these high elevation sites showed that despite decreasing sulfate deposition, atmospheric sulfate deposition, sulfate in runoff has actually increased by 300% over the past 30 years at the site where I'm working at Niwot Ridge, likely as a result of thawing permafrost and rock glacier features causing elevated pyrite weathering in this watershed. And so that elevated export of sulfate downstream could potentially be stimulating more methyl mercury in these downstream ecosystems. Additionally, these areas are experiencing increasing summer air temperatures, greater primary productivity, and faster snowmelt rate, all of which could be leading, and also changes in hydrologic connectivity, all of which could be leading to potentially greater methyl mercury production and total mercury methyl mercury export downstream. And already across the western US, there are over 200 fish consumption advisories just in the Rocky Mountain region alone, with over half those advisories over 1,500 meters in elevation. So at Niwot Ridge, I'm sampling a variety of alpine wetlands as well as dry meadow regions, which make up the majority landscape in this elevation zone. And then I'm also sampling subalpine wetlands below tree line, the inlet and the outlet, as well as subalpine forest floor, which again makes up the majority of this elevation zone. I'm sorry that the formatting is a little messed up on these slides. In addition to the wetlands, I'm also sampling streams draining from above tree line, the alpine stream, and then also below tree line in the subalpine stream. For the wetlands and dry regions, I sampled soil cores and then overlying water where present every other week during the 2021 growing season, which I then analyzed for these constituents there. And then I sampled the streams weekly for those constituents also during the same year. So to jump right into results, here I have soil total mercury and methyl mercury concentrations for the dry soil regions, the alpine wetlands, and then the subalpine wetlands inlet and outlet. And for both total mercury and methyl mercury, the alpine wetlands and subalpine wetlands are higher than the dry soil sites, not surprisingly. Only the subalpine wetland was significantly higher though. And I just want to draw your attention to the methyl mercury concentrations for the subalpine wetland were higher than we expected, all above 5 nanograms per gram throughout the growing season for a pristine high elevation subalpine wetland. And so to put these results into some context, I compared these values to past work that's been done in the Arctic, an area with similar environmental conditions, and then much more work that's been done looking at mercury contamination in the context of ongoing global change. And generally, for the Arctic tundra soils, they're falling around the same range that we see for our dry soil sites, whereas the methyl mercury concentrations are generally higher in Arctic tundra soils than our dry soil sites. And then comparing now specifically to wetland sites in the Arctic, you can see that my alpine and subalpine wetlands are falling around the range of values found in the Arctic, with my alpine wetlands generally lower and then the subalpine wetlands generally higher. And just note the Y scale for methyl mercury on the log scale for methyl mercury on the Y-axis, and those subalpine wetlands are on the higher end there. So this suggests that these subalpine wetlands in particular are acting as important regions for methyl mercury storage and transformations, and occurring at levels comparable to these Arctic wetlands, which are known for storing and transforming mercury on globally relevant scales. Next, to get at what processes might be driving those patterns we're seeing across this landscape, I conducted an incubation experiment with a lot of help from the Mercury Research Lab, and collected soils from the dry meadow regions, the periglacial salt fluxion lobe, which is one of those alpine wetlands, and then the subalpine wetland. In general, the dry meadow region, there's no standing water, but it does get wet from rain and snow events throughout the year, and generally has quite low sulfate concentrations. And then the salt fluxion lobe is permanently inundated, and it also has an ice lens underneath that holds water on the surface. And these sites have really high sulfate concentrations. You can get up to 15 milligrams per gram dry soil. And this is an example of one of those thawing permafrost features that I mentioned earlier. And then finally, the subalpine wetland is also permanently inundated and has much higher soil organic matter content compared to those other two sites and more moderate sulfate concentrations. So for each of these sites, I conducted an incubation experiment using a 201 isotope tracer to calculate ambient rates of mercury methylation potential. And then at the subalpine wetland site, I also did a sulfate amendment, 50, 100, and 200 micromolar, to see how this elevated sulfate export could potentially be impacting these subalpine wetlands. And what I found is that the alpine dry meadows, not surprisingly, have very low mercury methylation rates. This is an area that has really low organic matter and fairly oxygenated soils, so not super surprising. The salt fluxion lobe ended up having negative mercury methylation rates, and this is because we had some initial methylation after injection of the spike prior to that time zero sample freezing, and then that loss after that. And at first glance, this was somewhat surprising because these sites are low redox, fairly high organic matter, or moderate organic matter levels. But this is the site that has really high sulfate concentrations. So it's possible that that high sulfate is leading to a buildup of sulfides, which is then binding inorganic mercury and basically immobilizing it from further transformations. And I want to just throw out that this is just one of a variety of alpine wetlands that we use for this experiment, and there are a variety of others that have lower sulfate concentrations and maybe would have higher rates of mercury methylation, but were not included in this experiment. And then finally, the subalpine wetland had significantly higher rates of mercury methylation, which makes sense since this site has also the higher methylmercury concentrations. And this is likely due to the fact that there's high organic matter providing an energy source for microbes, and then there's a consistent source of inorganic mercury coming into the site through runoff throughout the year, and then more moderate sulfate levels. Again, putting these rates into some context, comparing to past work in the Arctic, the subalpine wetlands are falling within the range of rates that have been measured in the Arctic, but generally on the lower range, especially compared to peatland and wetland sites. And then this is the sulfate addition data here. So here are the ambient rates in the subalpine wetland, and these are generally lower than the 50, 100, and 200 micromolar additions of sulfate. Only the 50 and 100 were significantly higher, but this generally suggests that these sites are sulfate limited, and so continued weathering of pyrite and runoff of sulfate into these downstream subalpine wetlands could be stimulating greater methylmercury production in this region. So to take a look into what those potential downstream impacts are, I here have the methylmercury concentrations for the alpine stream, the subalpine stream, alpine wetland, and subalpine wetland inlet and outlet. And here found that the subalpine wetland outlet has significantly higher concentrations than the inlet, suggesting that methylmercury is being produced within this subalpine wetland. The alpine wetlands were generally lower than the subalpine wetlands, and suggest that these sites are not as important hotspots for methylmercury production. And this could be due to the fact that there is low organic matter. Additionally, these alpine sites in Colorado get really intense sunlight exposure pretty much every day. And so there's likely demethylation occurring of the methylmercury being produced in those sites and could be helping detoxify those areas. But these higher concentrations of mercury in the subalpine wetlands compared to the alpine wetlands does show up in stream runoff, where you can see the subalpine stream has higher concentrations than the alpine. All of the alpine sites were below detection. And this, yeah, so it suggests these values are generally pretty low, again, comparing to the Arctic. You can see that they're falling on the lower end of the range there. But there is still methylmercury in the system. And so I also explored potential implications for local wildlife at Niwot Ridge. And so I'll briefly show you those data as well. Some of that's still a bit of a work in progress. But up at Niwot Ridge was looking at the impact of food source and trophic position on a terrestrial, mixed terrestrial, aquatic, and aquatic food web. And jumping into these results, for the terrestrial food web, you see that mercury increases going from vegetation into pica. The concentrations are quite low, as we would expect for a terrestrial system, and low trophic position. For the mixed terrestrial aquatic, mercury increase going from the grasshoppers to caddisflies to birds and weasels. Not surprising, as we go higher in the trophic levels and also more feeding from aquatic regions, and we're seeing more accumulation. And then finally, for the aquatic system, seeing increases from the lake sediment to the zooplankton to the fish. And I'll add a little bit more on the, these are all of the organisms across, seeing we do have increasing accumulation with higher trophic position and more aquatic feeding. And to just show you a little bit more from the fish, I'm still analyzing more of the environmental data from these lakes. But these are the 4 lakes that I sampled across, and they're decreasing in elevation across this watershed. And as you move up in elevation and closer to the rock glacier input where that sulfate source is coming from, we are seeing higher concentrations of methylmercury in the fish, so potentially seeing that signal of sulfate stimulating more methylmercury production and bioaccumulation. And I'm getting to the point now of putting this work into a larger ecotoxicological context, and part of that is sampling dragonflies, which I did do this summer with mixed success. But I did find them at 2 sites, the highest elevation lake and then the subalpine stream. And so I'm currently working on weighing those out at the Mercury Research Lab, and we'll be analyzing them this fall. And that will allow me to compare this site to sites all across the US and put it within an impairment index that Colin and others have come up with to put this area into a broader context. So with that, here are my conclusions, and I'd be happy to take any questions.
All right, thank you. We good. Meet you halfway.
Make it a little easier. Hi, that was great. I was kind of curious about the wildlife context. I was really happy to see picas and some of these other species that were listed. So I was just kind of curious about some of your future efforts. Are there any efforts to relate, because there's a lot of related research on Niwot Ridge where folks are looking at survival or occupancy or for some of these wildlife species. So do you have plans to kind of look at your results and think about what that might mean for the wildlife health or behavior on Niwot Ridge?
Yeah, I have started looking into that a little bit. I might even have some extra slides. So yeah, just trying to get a sense of like what risk is actually existing for some of these organisms. All of the fish are below the historical EPA cutoff for safe fish consumption of 0.3 parts per million. So that's great. The birds are falling within a range of values that historically have been shown to have some reproductive or physiological impacts. This is a little tricky because depending on the species and the tissue being analyzed, it can be hard to compare across studies. All of my samples were 12-day-old chicks' breast feathers, and so I used like the equations that exist to convert to blood equivalent, but I think it can be a little tricky to say whether or not it's actually impacting those birds negatively. So yeah, starting to dive into that and hoping to talk to more folks who think about that more regularly.
Thank you.
Any other questions? Colleen? And I'll add the fish one, so they're below EPA thresholds, but they're right around where you start to see impairment in fish. So they're close to kind of, you might see biochemical impairment in some of those fish.
That's super cool, Hannah. I did my work up at Green Lake 4 decades ago, but really neat to see you there at Niwot and see some familiar lakes. I was just curious, so your incubation experiments, what month were they held?
What month were they in?
Held, yeah. Or was it over the course of the
So-
growing season? Or I guess I'm
Yeah.
curious specifically about the ionic pulse and like other water chemical parameters, like
Yep.
total nitrogen, phosphorus. Yeah.
Yep, so it was a pretty simple experiment. We weren't actually sure if it was gonna even work. The experiment I did was, I'll go back to those data, was 72 hours, and I'm only presenting the first 12 hours of data here because the response didn't appear to be linear over that time. So trying to figure out what equations are best to use. But this was basically a 72-hour incubation experiment, collected samples each day, so I have 4 time points, including time 0.
Got it. Like
and
in
then-
July or August or?
Yeah, it was in August.
Got it.
Yep.
Okay. Yep. Are you able, or does the capacity allow to look at other parameters too? Is there any concern about the pulse? I mean, I suppose in August, by then it's hydrologically flushed through.
Yeah, I do have time data or time series data for stream runoff, and can see that pulse pretty clearly occurring more in June.
Yeah.
Yeah,
right.
so I think at this point we were past that.
Yeah, right.
But that
Totally.
is, yeah, that's interesting.
Yeah, and so your dragonfly collections, just curious, are you collecting from Green Lake 4 or are you lower?
Yeah, that was where I found them, was Green Lake
No
4.
kidding.
Yeah, I was really
Well-
surprised.
I think Christy has a son who wants to help you collect dragonflies. Yeah. And I could probably assist too if there are any interest. And final question. So the fish, are they not stocked?
So this is sort of a funny situation. There are not actually supposed to be fish in these lakes, but they accidentally seeded the wrong valley. They did a flyover. And so these fish are now multiple generations. I think that was in the 90s. And they died out of Green Lake 4 because the lake gets too cold for them to spawn. But the lower lakes still have thriving populations of trout. But they're not naturally occurring there. Yeah.
Anyway.
Thank you.
Potential concern. Thank
Yes,
you.
no, absolutely. Any other questions? Colleen, any more questions? Thank you, Hannah, that was great. Okay, well, that concludes our session, and thanks everybody for taking the time to spend sitting here listening to all these talks, and appreciate all the interest, and enjoy the rest of the conference.